Thank you, Sandra and Liang, for introduction and invitation. So first, let me share my screen. Oh, okay, the screen was gone. Yeah, how about now?
Yeah.
Can you see the screen of slides? Okay.
Yeah, great, perfect.
Yeah, I'm so honored to speak at the data science seminar at Boston Children's Hospital. The topic is already introduced. It's integrative analysis of single cell. Today, it's not just that paper. I also include some more general background of GRN and some relevant things, but we will focus on that paper. We first talk about introduce the problems, and then mathematical modeling of GRN will be introduced, and then move to single cell, and then later applications. So during the talk, after each section, I think everyone, if there are any questions, feel free to ask. We don't need to wait in the end of the whole presentation. So we start from a differential expression problem. It's a very old problem in bioinformatics. So in a case control study, like between healthy and diseased, people normally ask what genes are differentially expressed between them. There are many computational methods and tools developed to address these problems. And a related problem to this is how are genes differentially expressed? So it means which regulators cause the differential expression. So if we have a good answer to the question 2, then it will help us to design a drug for the treatment of disease. It could be directly used for the therapy because it is more mechanistic. So if we look at the problem from the molecular biology central dogma, we know we have 2 copies of DNA. So how many RNA and how many proteins they were made is the problem here. So DNA will become RNA and RNA will become protein. And some of the protein is called transcription factors. They can have a DNA binding domain. They bind to the DNA and regulate gene expression. So those transcription factors, we say this regulates the gene here. And we have many of these transcription factors, and also many of them gene are also regulate other genes. So it will form a network. So in this network, we see the regulators can activate or repress their genes target gene expression. The color here represents the minus log 10 p-value of the differential expression. Of course, it's just a part of the network. So from C, we see many of the differentially expressed genes are enriched under a few number of transcription factors. So that means if we know the network, then we can identify which regulator is really causing the differential expression. This regulator should be the potential driver regulator of the disease. But here the inference of GRN is a very hard problem. So first one is it is a mathematical way of looking at the problem. Let's say we have a few regulators, transcription factor 1, 2 here, and a few target genes. Then the inverse, the GRN is often modeled as an inverse problem of ordinary differential equations. Let's say the production speed of gene, like gene I, is modeled as production speed minus the degradation speed. The degradation speed is like have a direct ratio of their expression level. If their expression is higher, they will degrade faster. Then the production speed is a function of many regulators. Like we know the expression of those regulators, or should be activity of these regulators. And then if we know which regulator regulates the gene, that's the network, it's called Ri, and a function of those. So what we observed is the transcription factor and genes paired expression on steady state that dg over dt is 0. We only observe these. And then unknown is we don't know the regulator of gene. What are the regulator of gene? That's the network structure we don't know. And also we don't know the function that determine the speed of GI. So these two are unknown. So this mathematically the inverse problem is very hard because it only observes the partial data. So historically, many of the case, the Fi is modeled as a linear function. Then it is, I'm gonna talk about this is the over simplified case, so it will underfit the model. So from the biology perspective with GRN, it's a very fundamental problem. So first, if you look at the developmental biology, people are looking at like asking why so many different cell types looks or they function so different, even they have the same DNA sequence. They are from the same cells. They're supposed to be same, but they looks very different. What makes them different? That is called, it should be the cell type or context-dependent GRN controlling them. Then if you look at the genetics, many cases like non-coding genetic variant causing disease. How a variant in non-coding region could cause disease. That is a genotype-specific gene regularity network. And if you look at many medical health researchers, they're asking how a healthy cell responds to the environmental signal and become from healthy transfer to disease, or how the therapy of drug can convert the disease case back to the healthy. That is actually an environment or drug dependent gene regularity network. So all these cases, it could be summarized like context-dependent GRN. The context could be cell type in the first case, it is genotype in the second case, and the environment for the third case. And the GRN is also the core of the precision medicine. The reason is people have different genetic variant, and then because of the genotype is different, of course, their gene regulatory network in each individual, in each cell types are different. Because of the network is different, even the same environmental factor like drug or food or like lifestyles, they cannot change different set of genes for different people. Of course, then the phenotype is different. That is the framework we can think the precision medicine way. So how to do the personalized GRN is an important problem. So let's look at the previous studies. Like there's the benchmarking of GRN by experimental data. This is from 2020 Nature Biotechnology Paper, benchmarking algorithm for GRN inference. So in this paper, the benchmarked like many GRN methods using a few data sets which have ground truth data. The values here are using the AUPR ratio compared to a totally random prediction. So 1 means totally random, 1.3 means 30% more accurate than a random prediction. So if we find 10 genes in a random prediction, and in those methods can identify 13, then it's going to be 1.3. So in many cases, we see their AUPR ratio is like, many of them is less than 1. Some is around 1.0, some are 1.1. Sometimes in some data, it can reach 1.3. That's the maximum case we can get from the from computational inference, which means overall, for over 20 years of study, the GRN inferences keep improving, but it is a very hard problem. So when compared to the ground truth data, it is like very low accuracy. Then why the GRN inference is so hard? A simple answer is the problem itself is too complex. So if you look at a single gene, they are regulated by non-coding elements like promoter, enhancer, insulators, like many different type of regularity elements. We call it cis regularity element, CRE. And then transcription factors or other proteins can bind to the CRE and then make a looping to the promoter or other enhancers to regulate gene expression. So here, first the problem is like non-coding regions involved in gene regulation. Many of the models only using gene expression, so that is infer a high accuracy GRN is very hard. Second, they have a nonlinear interaction among transcription factors, nonlinear interaction among CREs, and also interplay between TF and CREs. That makes the problem so complex. So the GRN we're talking about is not talking about the co-expression network and PPI. The co-expression network is a very common network people use. That is, the expression of genes are correlated or not. This is actually the consequence of the GRN. If two genes are regulated by same factor, like if they are siblings, they're more likely to be co-expressed. So not the parent-child relation, this is a sibling relation, more likely. And PPI is the interaction among regulators, it's physical interactions. And the GRN we're talking about here is like three types of node, like TF, element, and gene, three. And they have three types of edge. TF regulate RE is called chromatin regulation. So we cannot have direct measurement of this, but we can have a physical binding of TF, like ChIP-seq data can give us which TF regulate, which TF bind to which RE. Like binding is a foundation. Like if they do not bind, of course they don't regulate. But binding doesn't mean it really regulate. Even you perturb the transcription factor, we see no change in the RE. That is very common. And then this regulatory network is from enhancer to gene, RE to gene. These are cis. And we have some relevant observation. It's like looping. 3D genome, like high-C data can give us which have loop. This is the foundation. No looping means no regulation, but looping doesn't mean the real regulation. Second one is EQTL, between the genetic association between genotype and expression. This is the consequence. If they regulate an RE regulate gene, and if there is some genetic variation, we could observe EQTL. But in some cases, you didn't observe EQTL, it doesn't mean they don't regulate, because many of the regularity element is highly conserved, and we never observe any genetic variation. No variation, so that no EQTL. And transregulatory network is from TF to gene. Also, we can do perturbation experiment or ChIP-seq. So perturbation experiment is constant and ChIP-seq is foundation. So we cannot directly observe this, but we can some are sufficient, some are necessary, but very hard to find which are sufficient and necessary. So the challenge in this context-dependent GRN, first the data is incomplete. It will be very hard to get both deep and broad data. By deep, I mean like ENCODE project. For some cell type, we do gene expression, ATAC, histone, many different type of transcription factor, ChIP-seq. This type is like very deep, right? Even single cells. Some data is like deep, but some data is broad, like GTEx. They do many people's, or like GWAS, they do many people's genotype and phenotype. But this data is like do not have deep multi-omics. So it's very hard to get both deep and broad data. And GRN inference from single modality data is like just slightly better than random. So it needs a system biology approach to integrate all pieces of data together. And we also need a causal and mechanistic model. We not just require association. That makes it even more difficult. And also the limited ground truth data. We have very limited number of real ground truth data. So if we have large amount of ground truth data, we can use the machine learning or the large language model to train those. But now we don't have large amount of training data, so we cannot this way to address the problems. So talk about the GRN inference from genomic data. Once we have expression data, gene expression data, people can do TF and gene network. but it do not include the non-coding region. So its network is like very incomplete. Then some protein DNA interaction like ChIP-ChIP or ChIP-seq can do TF's target gene and target RE. But this one is like every experiment you only do one single transcription factors. And later people have chromatin accessibility data, which is can identify which part of the genome is functional. Just looking at like the chromatin is like very highly packed, like total length of genome is about 1.8 meters. To pack them in a tiny nucleus, they need to be really tightly packed. And some parts are open that could be captured by ATAC-seq or DNA-seq. And that open part, it could be identified by transcription factors, and why factors can bind here. So ATAC and DNA-seq data can give us this information. Then paired, like from the ATAC or DNA or single cell ATAC, people can know, okay, which elements are open in non-coding region, and they are binded by which transcription factors. Because we know this transcription factor sequence preference, we know which factor will match to this DNA sequence. We can identify those type of network, but no gene, and also cannot distinguish TF from same family because they share same motif. Then later we have single cell multiome, then it's gonna even more richer data. It observes both gene expression and ATAC-seq on the same cells. So how to infer this network among the three types of node? It's the problem we're going to introduce later. So the summary here is GRN is required for driver identification, and the content-dependent GRN is very fundamental and key for precision medicine. We talked about three components of GRN and three types of node and edge, and we talked about the challenge in this GRN study, and we also talked about RNA plus ATAC-seq together for single cell or bulk. It's a great combination for the GRN study. So if anyone have questions at this stage, can ask, and then later I will move to the more methodology part. No, it seems okay. Great. So let's move.
Sorry, we do have a question.
Yeah.
From Age. Let me just unmute Age. Okay. All right. So I think you can unmute yourself. You can speak now.
Hi. Nice talk. I had a question. Say if we have RNA-seq and ATAC-seq data from a patient, will we be able to get the gene regulatory network for that patient?
You mean you have RNA and ATAC data from a patient, right?
Yes.
Yes. So yes, we can infer their GRN. We have a few ways to process. One is like if you only have one data from one patient, we can infer a network for that patient, right? Another way is like if you have many, like some diseased people and some healthy people, we can put everything together and train a network, and then see which regulator is differential among the diseased and healthy based on the network. Yeah, we can.
The second part is clear to me. If we had deceased and if we had normal, then we can identify probably differentially expressed genes and probably differentially expressed regulators and GRN. But if it is from just one patient, will we be able to, I mean, my question is more theoretical. Can we identify which are the regulators that are causing that patient disease if we have data only from one patient? Mathematically, is it possible to do it?
Yeah, so if we using, I'm talking about single cell.
Yes.
If we have single cell data, we can learn the across cell variation, then still can capture those things. So even from single patient, we can do that.
So then are we assuming that we have normal cell and then cells from the normal tissue and from the diseased tissue? Do we have to make that assumption?
Not necessarily, because in normal case, like when you're collecting those cells are a mixture of many different cell type or cell states. They have some variations. So from those variations, we can learn their association.
Okay. Thank you. Thank you so much.
Other questions? Okay. Okay, so the application of the differential expression or differential activity part will be discussed in the end of the study. We have some applications that will make it more clear. Good, thank you. And let's move to the second part. It's the GRN modeling. So first, let me briefly introduce this paper I done 2017. It's about almost 8 years now. That is called paired expression and chromatin accessibility data. So in this paper, we ask 3 questions, like how are genes expressed? The simple answer is transcription factor and active RE control the gene expression. That is based on bulk data. And then how RE get activated? We model them as like open chromatin, and the chromatin regulators binding would affect the RE activation. Then the third question is the chromatin regulator is not like transcription factor. Transcription factor have specific sequence they recognize, have DNA binding domain, but the CR chromatin regulator do not. So we ask a question, how CR bind to DNA? We model them as TF and CR have protein-protein interaction, and transcription factor will recruit CR to bind to DNA. And we use some knowledge and of prior knowledge and fit those models. So basically, input data is RNA and attack from 25 mouse tissue and cell line. Later, we also did it for hundreds of human tissue and cell lines. We use the knowledge of PPI. We also know the knowledge of TF and motif matching and their physical distance in enhancer and genes. Then the blue node is gene expression, orange node is attack, and the gray node is latent variable. So here we have the model is like maximizing the distribution, conditional distribution of target gene given TF, TR, and openness. The third equation is addressing the target gene expression fit by TF and active RE, but active RE we don't know. The second equation is addressing the active RE by open chromatin, CR expression, and chromatin regulators binding, but we don't know the chromatin regulator binding. Then the third model is like modeling the chromatin regulator binding from TF and open chromatin. So these 3 equations, we fit them jointly, and output will be the parameter at different levels, like TF to CR interaction recruitment, CR to RE with an alpha parameter, and RE to TG is the beta parameter here, and TF to TG is the gamma parameter here. We learn this different level of networks, and also we learn the latent variable of CR binding and RE activation. Then we apply this Pekka model for 2 cases. One is identify the causal warrants for high altitude adaptation. Another one is a master regulator for a differentiation problem. Let me simply go over them. So this model is the Tibetan people is living in high altitudes and for long times. So after the natural selection, these people already adapted to the low oxygen environment. But normally people could travel to the Tibet area. They will have many heart-related issues, or if the pregnant woman travel there, they have a very high risk of doing the fetal loss during pregnancy. So if you look at from genetic data, we know the EPAS1 is the very important regulator here. The EPAS gene is normally low expressed in Tibetan people than normal lowlander Han Chinese, and these people, because of the low expression of EPAS1, they can better adapt to the high oxygen environment. And we used the network study and identified the regulator of EPAS1 is called HIF1 alpha. That is this factor is like respond to the low oxygen very quickly. The HIF-1 alpha factor is binding to those enhancers of EPAS1. This is the HIF-1 alpha motif. This will recognize GCGTG. That is the motif we recognize. But in Han Chinese, their sequence is like T, G, C, G, T, G, C, C, A. But in Tibetan, this C is changed to T. Because of this word, this T, the HIF-1 alpha cannot recognize this base pair. They do not bind here. So it means in Tibetan people, this regulation is disrupted. So because of that, even in low oxygen environment, the EPAS1 experiment cannot go up very quickly. Then we do some experiment to validate this. Indeed, if we change the word from C to T, the gene expression will decrease by 1.8-fold. So that means this edge is validated by experimental and can explain why the Tibetan people can adapt to the environment. The second application here is the epidermal differentiation. We have different age, like times, like day 0, 7, 14, 21, 43, and later. That is the stem cells grow in certain medium, and after about 50 days, it will turn to the epidermal, the mature keratinocyte. It's a skin cells. So here, at each time point, we have RNA-seq and ATAC-seq. Then we infer the networks. So that is the network of the earlier stage. So in the network, we identified the TF52C is the largest node here. It's the most important node in the network. Then if we do the experiment, like if we overexpress this gene at this D0, then after 7 days, it becomes the surface ectodermal progenitor without using any other those mediums. So only one gene can replace those mediums here. And the second stage is the maturation stage. They also have the network. The most important gene is P63. And if we knock this gene down, we cannot get epidermal. Then it is consistent with the experimental validation. And so we identify the master regulator of the maturation stage as well. So in the PECA model, it's like we consider non-coding DNA and chromatin regulator, and it is a mechanistic model for gene regulation from RNA and ATAC. And application can identify a regulator and causal water and enhancers. So what is considered and what is not considered in that model? Considered is we consider the non-coding region that is previously in model do not consider, and we also incorporate the knowledge like motif of protein and DNA bindings. What is not considered is that is bulk data, so it's not considered the mixture of cell types. Normally, the tissue is a mixture of many different cell types. You see some expression change, you don't know if it's really expression change or it's just because of the proportion change. And another thing is the complex. The system itself is so complex, we require large sample to fit. The data here is not enough. Then the single cell multiome data is from 10x Genomic or like many different technologies now, but this one have commercial cases, so it's widely used. At each single cell, they profile gene expression and ATAC-seq. So we can easily get about millions of cells have both RNA and ATAC-seq available on individual cell level. That is the ideal data for learning gene regularity networks. So here is the paper we published last year. It's learning GRN from single cell multiome data and also leveraging the external data sets. This method is called LINGER. It's lifelong neural network for gene regulation. So the LINGER framework is the input is we have a gene expression and chromatin accessibility for individual cells, like 2 data matrix. Each column is 1 cell. Each row is 1 transcription factor for the first matrix, and second matrix is for each row is 1 regularity elements. And we also know their interactions as prior knowledge of which motif bind to the which influencer. Then the model itself is just a simple two-layer neural network. The output layer is single node. It's a one target gene expression. That is one gene, one model. For one gene expression, we have regularity element within 1 million base pair of that gene, maybe about hundreds of regularity elements and hundreds of transcription factors. Then the first layer is we interpret them as regularity modules with weighted sum of TF and REs. Then in this weighted sum, we know which transcription factor have binding to which RE. We have a graph, known graph. We encourage their interactions by using manifold regularization. It means in this module, the activated nodes tend to have more interaction in the graph. Then it's a regression problem of the gene expression. The gene expressions we know and inputs we know, we learn this regression model. And then after learning the model, we use a Shapley value, which is from an explainable AI technique from game theory, to learn which node, which input, like which transcription factor and which RE are important for the gene expression. Based on the Shapley value, we know which TF and which RE is like regulator of this. But the problem here is the model is like only training on single cell data is not enough variation, because the single cells, even we have millions of single cells, they are like maybe very similar to each other. So the variation space is not that big. To use this, we use a pre-training strategy, like similar to the foundation model and fine-tuning strategy here. We train the model on bulk data across diverse tissues from ENCODE project, like hundreds of tissues are included here. We train the same model and we save all the parameters, and we also save their confidence. That is the way we call it lifelong learning. It's different from the current foundation model or application, like detrain a foundation model and fine-tuning in the other data set. Here we use the better way, like we not just save the parameter itself, we also save their confidence, like feature information of the parameter estimation. If we are very highly confident about the parameter, then those parameters cannot change too much in the single cell data. If some parameters, the feature information is very low, that means this parameter, even you change the parameter a lot, it's not very sensitive to the results on the bulk data. So in this case, if we changing the parameter and get a more decreased loss in single cell, this parameter will be changed. We add this elastic weight consolidation. The Fi is the feature information, and the distance between the bulk data parameter and the single cell parameter. So if these two how different they could be depend on their feature information. So here the distance we didn't use the Euclidean distance because single cell data and bulk data have their scale is different. So we use cosine distance, cosine similarity as the distance here, then they will address the scale issues. Then the benchmarking is we use the PBMC data of 10,000 cells, and we also use in silico mixture of 4 different cell lines. Then ground truth is we have known ground truth data for ChIP-seq, promoter capture high C, single cell EQTL, and knocking down data. For those, all the available data, we downloaded them and take them as ground truth. And the evaluation metric we calculate AUC, area under curve, and AUPR ratio compared to a random predictor. Then the result here is like TF and RE network. The orange one is our curve. In AUC, we get 0.92. It's better than previous methods a lot. And in the ChIP-seq data, we also did much better job compared to the simple method and the best method in previous studies. And in promoter capture height, they give us an EQTL can give us the enhancer and RE and gene connection, and our AUPR ratio is like much higher than the existing methods here. And even in EQTL estimation hours, like much higher AUPR ratio, we can give about 6 in the EQTL based validation. So here is the TF and gene based. That is like many methods here because both are genes. In this case, like linear AUPR ratio is about 2.25, which is much higher than the previous method, which is about 1.2, 1.1. That is consistent with the previous benchmarking papers. So this is like previous method is about 17 to 29% above random prediction, but we do is like 2.25 is 125% above random prediction. It's about 4 to 7-fold relative increase. That get a really increased accuracy in gRNA inference. So summary for the method is we use RNA and attack data to train model gRNA. And for PECA and Linger is the single cell version of RNA attack to learn gRNA. Then innovation here is first we use lifelong learning, which is actually a Bayesian style transfer learning. And we incorporate the knowledge using manifold regularization, and we use the game theory concept Shapley value to extract the importance and infer the network. Then the result is like we improve those results significantly. Okay, then I go to the application quickly. So we test for each gene, we know it's regulated by transcription factors and also regulated by enhancers, cis, so which are important. So for many of the genes, if you look at the Shapley value of them, they are similar. But for some genes, like about 4% of the gene is more cis, more enhancer, like cis nearby region dominated. About 2% of the gene is about transcription factor dominated. Then we want to see what's their difference. So first we check a gene set called selectively constrained gene. This set of genes are like highly conserved across all human populations in their protein coding region. So that gene should be relevant to the human survival. And this type of gene are highly enriched in like more enriched, like about 3-fold more enriched in trans-dominant gene compared to cis-dominant. Why we care about this is because a previous study showing the EQTL study and GWAS, it's a disease study. So that GWAS study, GWAS gene are more enriched in the selectively constrained gene, and EQTL is not. So this study is consistent with them. That means to identify the disease gene, we cannot just rely on EQTL. We have to focus more on the trans regulation, not cis. And here's one application of IBD. The data we use is differential express gene from bulk data. And we also use the GWAS gene from the GWAS study called DAG. And we have the single cell multiome data from one healthy donor to infer the network. So in the network, first ask which cell is more important. So many disease-associated genes like GWAS gene should be enriched in the target gene of TF, like controlling important TF. So for the different cell types, those are different. So like, for example, in the monocytes, the important transcription factors are regulating more GWAS genes. That means monocyte is more relevant to the disease. And here, that result is like 3 types of monocytes are more those transcription factor, like important transcription factor and bottom ranking transcription factor have very different GWAS enrichment. Then we also do one interesting thing we find is the differential expressed gene and GWAS gene do not have a very good overlap. But if you look at their upstream regulators, they are highly correlated. Like their differential expression, each dot is one transcription factor. Y-axis is there. If their target on average is differentially expressed, then if x-axis is the score we defined is based on the importance of the transcription factors. That means the DAG, disease-associated gene, and differential expression do not have good overlap, but their upstream transcription factors are highly consistent. That indicate a feedback mechanism between GWAS gene to these genes through the transcription factors. And the last application here, if we go back to the differential expression case, how the genes are differentially expressed. So here the concept is called transcription factor activity. In the left panel, their transcription factor is expressed but not active. It's just in the cytoplasm. In the right panel, the transcription factor is expressed, making a protein, then they go back to the nucleus and bind to DNA. In this case, the transcription factor is involved in gene regulation. In this case, it's active and expressed and active. This cannot directly profiled from using experimental technique, but we can estimate it using the linker infer GRN. So we have one healthy donor multiome data. We run linker and get the network. Now we have from single cell or bulk from large cohort only gene expression data. We can estimate the transcription factor activity based on their target gene. For each transcription factor, we can average their target gene expression, then we can get the activity. Then we can, for each individual, we have transcription factor activity, so we can test between diseased and healthy if their activity is different. So here's one study, in bulk microarray data from healthy and AML cancer, then we see no expression change for this transcription factor. But this one is the positive control because it is in many experiments have shown that it is a cancer suppressor. But if we compare their activity, so we only using one single cell multiome data in infer network and calculate their activity. The activity is significantly different, and in TCGA data it also shows significance. That means we can estimate that activity. It's not detected by expression, but based on activity, we can get that. Another example is like in TCR stimulation in CD4 T cells of 8 hours versus 0 hour. So these two genes have, if we test their expression, no significant change are observed, but in activity, they are very different. And this is validated by the post-proteome data that can give us which transcription factor is active. So this one is both these two is like in the post-proteome data, they have show change. That means their activity is changed, but expression is no change. But based on the network, we can identify those as the differential regulators. And here is a study of alcohol use disorder. We have 163 individuals healthy, 82 healthy, and 82 alcoholism, and 81 healthy. And we infer their networks, and we tested like many factors are differentially active. And among them, those error one is validated by drinking-based GWAS study. That may be the potential driver of the alcoholism. Really last one, I want to talk about a little bit about spatial application with our ongoing project. We have single cell multiome, each cell has peak and gene, these two metrics, and spatial transcriptomics, each spot, we have gene expression data, but we don't have the peak data. So using these three, we can estimate what is the peak data in spatially. And we can estimate this well based on the variational autoencoder framework. And if the gene is more spatially variable, we can estimate even better. And then based on that, if we do a crown-wire motif scanning, motif twin in raw, these 3 factors motif have this pattern on spatially, and our predicted one is highly consistent with the just transcription factors, this motif activity. Then another case is like, that is the crown-wire motif activity. Motif activity is for the TF from same family have same motif, so we cannot distinguish among them just based on the motif. Just only from technical data we can do this. But if we do the regularity analysis, we know each transcription factor, we know their target. Based on that target, we can infer the activity, what we talked before. Then we see this 4 transcription factor, their activity pattern is different in spatially. But if you only look at the crown-wire or the motif activity, these 4 things are merged together. We don't know which area is from which transcription factor active. So let me summarize here. We talked about the GRN and challenge. We talked about the methodology for GRN inference, and also for single cell and with validation. We have application of GWAS and differential express gene using GRN. We have the TF activity comparison and to identify the hidden drivers. And we also talked a little bit about spatially resolved GRN. And I acknowledge my collaborators and my NIH funding and my lab members. The linker is done by Kaya Yuan. She's now an assistant professor in South Tech University. And the spatial is done by Ishita Tepnes. She's currently a graduate student in my group. Thanks everyone for listening. I am happy to take questions.
Great. Thank you so much for a great talk. It's very interesting talk. Okay, do we have any question from the audience? Okay. All right. AJ, you can unmute yourself.
Very excellent talk. A lot of nice tools and great applications. One quick question I had was, how many transcription factors do we have a motif known? I mean, how many, if the motif is not well known for some transcription factors, can it still be used?
Yeah, in total, the number of transcription factors is about, it have likely different definition, but people think about 1,600. And among them, about half, 800 of them have known motif.
Okay.
Yeah. The other part also must be involved in gene regulation. They don't have motif. It's like their motif is keep changing in different contexts. So that is make it complex. So yeah, that's another part, challenging part in GRN.
So with the tools that you have, it's mostly focused on transcription factors with well-defined motifs. Is that true?
Sorry, can you repeat the question?
The tools that you showed today, the Liger and the other associated ones, they are focused on transcription factors with well-defined motifs?
Yeah, they only focus on transcription factor with known motif, like 800 motif transcription factors.
And these motifs you have taken from, and these motifs you have taken from ENCODE, or you done your own ChIP-seq and other?
Yeah, that motif is like basically collected from different database. Like ENCODE is one big part of them, Jasper, Homer, Transvac.
Thank you so much. Excellent talk. Thank you.
Great.
I have another question. Hello, Dr. Duren, and thank you very much for the great talk. So actually, I have a question more about a kind of modeling the dynamic gene regulatory network, whether or not linker currently or potentially be able to incorporate pseudo-time or infer some trajectories to kind of model more dynamic GRNs.
Yeah, linker is not including the dynamic GRN, the pseudo-time result one is not including. Previously, we have done a real time-based GRN. It's called TimeReg. In 2020, published in Genome Research. That's for bulk data. And then now our lab is currently working on the dynamic GRN from single cell data, like similar concept using the pseudo time.
Thank you.
Great. Chonghui, go ahead.
Thank you, Professor Duren. It's a great talk. So I have a couple of questions related to the model training part. So you mentioned about like using the RNA-seq and ATAC-seq bulk data for the training. I just wonder, like when you train the data, are you using like all the samples, for example, like from different diseases to build a more like general model, or you are building like, for example, the disease specific like models when you do the training?
Yeah, that's a good question. So we didn't include disease sample because that is only from the ENCODE. Basically, most of them is from ENCODE project. So we're trying to capture the variation across the different human tissues and cell types. And then because the concept of lifelong learning is like, once you train the model, you can keep updating. You can keep updating the results multiple times. So first layer we have from all the human organs, we train one model. And then if you have data for a specific disease from many patients in bulk level, you can retrain them. And then after that, if you have single cell data for a few patients, you can refine them again. That's the concept of the lifelong learning. Let's keep training and improve, do not lose too much yet. Okay, great. I just wonder, like, how long does it take to, train once for using? Just wonder, like, what is the like the sample size you're currently used for training, and how long does it take to perform one training? It's not too much, because the model itself is very simple, just 2-layer neural network. But it has many models because each gene we have to train separately, about like 20,000 protein coding. We're doing that. So the ENCODE data have about 200 of tissue and cell types. So for that training is about like 2 days. It's not super long. And once you having that one, training your own data, normally like within 24 hours in single cell, we can solve that problem. Okay, thank you. So yeah, one last question. It's basically regarding the TF. If I understand correctly, you're using TF the expression level of the TF as the input, and you also briefly mentioned about like the TF activities, which you use basically the invert target like gene expression to basically calculate the overall TF activity. So just wonder, like, can you explain a little bit how to understand, like, you use the expression as the input data, because I understand that it basically is to model TF activity using expression, and then but the outcome you are saying using the target DEGs, like target genes as the activity. That is the main reason makes them different is because of the motif data. The transcription factor have motif binding to those genes, and using that information, you know which one is their downstream target, right? And then their average is more reflecting their activity. Okay. Yeah. Thank you.
Great. AJ, I think you have another question.
One quick question because of this. How do you compute the activity? I mean, I understand transcription factor as a motif to the target gene, but how will I know the activity of my TF based on the expression of the target genes, going up or down of the expression?
Oh, you mean how to estimate the TF activity from target gene expression, right?
Yes.
It's basically a weighted average. You know the target gene expression and which one is more likely to be their target, and then do a weighted average of the target genes. So
if the expression goes up, then the activity is supposed to be high, and if the expression is lower, the activity is lower. Is that true? Is that how it is?
If activity of majority of, if expression of the majority of their target genes going up, then we say activity is going up. If majority of the target gene is going down, then activity is going down.
Okay.
Not based on the TF itself expression.
Okay. Now it's clear. Thank you. I mean, there is something called Viper, which also has something similar. Have you compared how that works and this works, or is it just that this is working well for you?
You mean the TF activity?
Yeah, there is a tool called Viper from Califano Lab, which also predicts the activity. It also
the activity part is like very hard to benchmark.
Oh.
Yeah, we don't have really ground truth for the activity estimation. In the application I showed two examples, like very few number of transcription factors have those possible correlation data. Then those one is like, you know, the available data is so less, it's very hard to systematic benchmarking.
Okay.
Great. Right, Tammy, I don't think we have more questions. All right, maybe you can stop here. Thank you so much, Dr. Duren, and I hope you have a great day.
Thank you all. Bye-bye.
Bye. Thank you.